technology, the internet, GPS in the palm of your hand, autonomous operation.
Technology is a driver of our times. Since its founding in 1958 in the midst of the Cold War, DARPA, the Defense Advanced Research Projects Agency, has been a driver of technology. Welcome to Voices from DARPA, a window onto the history, people, and accomplishments that have earned DARPA membership among the most admired places and cultures of innovation. My name is Ivan Amato, and I'm your DARPA host. In this episode of Voices from DARPA, we will take a tour of an ambitious 10-year project to merge biology and engineering into one of the most powerful engines of molecular invention the world has known. Although some work still remains to be done, the program now is winding down to a close, but not before its research performers already have delivered a new and versatile technology platform whose consequences have begun to ripple out. New companies, follow-on investments, chemical and materials-based technologies for the Department of Defense, and perhaps one day for the public at large. The payoffs already have begun to accumulate.
Biology can do things that no other technology can come close to doing. It can replicate itself. It can adapt. It can evolve. It can scale from 1 to millions to billions effortlessly in a day. And it's programmable through the genetic code. Biology is technology.
That's former program manager Alicia Jackson in 2015. She was speaking at a meeting that showcased the mission and projects of the Biological Technologies Office, or BTO. She had been at DARPA already for several years and was preparing to hand off stewardship of the Living Foundries program. She launched the program in 2011. By 2013, an extensive set of academic and industry performers, that's researchers in DARPA speak, had begun work to realize the BTO vision that biology is technology. Living Foundries was just one of the more audacious embodiments of that R&D mantra. Jackson argued at the 2015 BTO showcase that biology is the pathway to advanced new technologies that will empower us to make useful chemicals and materials like we never have been before.
It offers us a chance to combine all of the advances that we've seen over the past 50 years in electronics, in physics, in computer science, in data analytics, and combine it with the last 2 decades of advances in genomics, in biology, and neuroscience, and come up with a technological capability that far surpasses anything that we have today. It would be truly astonishing to have a technology that could leverage both of these aspects from the engineering world and from the biological world. It could offer us solutions and capabilities that we just don't have today.
Former program manager Renee Wegerson, now at Ginkgo Bioworks, picked up the baton on the Living Foundries program in 2017. In a Voices from DARPA podcast episode in 2018, she shared a status report on the project. It was a pivotal point in the project, whose first phase was known as ATCG. That stands for Advanced Tools and Capabilities for Generalizable Platforms. This was the phase, said Wegerson, in which the Living Foundries inaugural program manager, Alicia Jackson, intended to bring biology and engineering together in novel and ever more empowering ways.
She had this vision to start to create a tool bed where we could distill biology to an engineering practice, where you could take something like the design, build, test, learn cycle of engineering and apply that to a biological system like a microbe, an E. coli or a yeast or other type of bacteria, to make products and molecules that would be of value to the DoD. And I really think that her inspiration came from looking at biology as this incredible system that could make a real diversity of molecules that extends well beyond what we can make through traditional manufacturing and chemical processes. So when you have access to that diversity of biology, can you start to manipulate that to make new molecules, things that have never been made before by biology, which are some of the most ambitious goals of the program. So her foundational investments really created the tools under each one of those categories, the design tools that you can quickly, in an automated way, design a biosynthetic pathway to stitch genes together to put into a microbe. Build, so this is the DNA synthesis and insertion of DNA into a living system, testing. This is the mass spectrometry and other measurement tools to look if a product's been made, and importantly, learning. So this is some of the designs you make are not going to work, and programming that into your learning cycle to not use that design and make a better design next time is really important. The first phase of the program was to build those individual components and tools, and now the second phase is to really integrate them as a platform to create this foundry type capability that the U.S. military or a customer, a business customer, could go to a foundry and say, I would like you to make this molecule. Can you develop the biosynthetic pathway and the organism to do it? So the ultimate demonstration of that capability, that foundry capability, is to make 1,000 unique molecules. And we're 551 molecules in.
That was in 2018. The team of performers that were then working their way up to and now beyond the 1,000 molecule mark include researchers at the biotechnology firms Amyris and Zymergen and their academic colleagues at the MIT Broad Foundry. Each performer group brought different strengths and approaches to the table, says Ann Cheever, the BTO program manager who is now taking Living Foundries to the finish line.
So they all have different strengths and different approaches. One of the strengths of the MIT Broad Foundry is in some of the early sort of development and analytical approach to determining some of the biosynthetic pathways. So their goal was never to scale up. They're not a commercial entity, and so they were very good in sort of the very methodical approach to understanding what kind of biosynthetic pathways would go into creating a molecule. Amyris as a commercial entity has been very successful in scaling some of their products. And I think some of the early development under ATCG has definitely aided them in that path. And Zymergen, I would say, in terms of molecules and their commercial approach, has been very good at developing new materials. So using certain polymer monomers, for example, and thinking about new materials that can be made with that.
At the heart of all this biosynthesis and ultimately larger-scale biomanufacturing is the process of fermentation in microbes.
That's the actual sort of liquid tank where you put all your microbes and where you make them happy and you feed them and they can produce a molecule. And then from there, there's the purification of that molecule and separating it from that liquid and those actual cells that produced it.
Just as brewers and bakers rely on microbes to convert sugar into alcohol and to generate carbon dioxide to leaven bread, performers in the Living Foundries program genetically engineer yeast and bacteria to ferment or chemically transform different kinds of starting molecules into more useful molecules and materials. Some of these transformations require the scientists to install a number of genes that govern the in-cell creation of entire biosynthetic pathways, the equivalent of a new tiny factory inside the cell. That's why the relatively new term synthetic biology applies here. And because it now is possible for researchers to mix and match thousands of genes into an infinite variety of biosynthetic pathways, the potential to turn microbes into living foundries for churning out molecules with different traits and uses also seems to have vast potential. In recent years of the program, researchers have produced, among other useful materials, antibiotics, drug precursors, pesticides, adhesives, fuels, filtering materials, mold inhibitors, detergents, catalysts for chemical reactions, and optical materials such as liquid crystals. Synthetic biologist Christopher Voigt has been a principal investigator on the project at the Boston-based MIT Broad Foundry. I asked him, as a leader in the synthetic biology and biomanufacturing arenas, to record what he thought Living Foundries added to the cause.
Living Foundries was unique in its focus on improving the foundational capabilities underlying engineered biology. In other words, getting better at genetic engineering for the sake of genetic engineering, without a particular application in mind. As a result of this, projects that were impossible in 2013 are trivial today. You see the fruits of this work across DARPA programs that are bringing specific operational capabilities online, from harnessing living cells to build infrastructure, to converting plastic waste to food, to creating new domestic sources for rare earths, as well as countermeasures to handle the next pandemic. The cells being built for these applications use parts and methods from Living Foundries.
Voigt is referring here to a range of DARPA programs. Engineering Living Materials, or ELM, a program known as Resource, one known as EMBER, which stands for Environmental Microbes as a Bioengineering Resource, and one known as the Pandemic Prevention Platform Program. All share elements with the Living Foundries program.
At MIT, we use these methods to build over 1,000 new molecules in the time period. These include precursors for energetics, monomers for resins and polymers, targeted antimicrobials, absorbents, optical coatings, obscurants, and so on. One lesson learned is that the rapid scale-up of a biological technology remains a critical challenge. We can now rapidly engineer cells, but moving from a few milliliters to millions of liters is challenging and slows implementation.
When Anne Cheever took the managerial baton over from Renee Wegerson in 2020, she knew it already had taken a lot of work for the program to achieve what it had achieved, and she knew it would take a lot more to do what still had yet to be done.
So there were a whopping 16 initial performers under ATCG, and they had various projects for varying lengths of time, that ranged from academic labs all the way to institutes and companies. They really spanned the gamut of approaches. They all had unique approaches and strengths, but overall, it was their collective efforts that really kind of set the stage and transformed that fermentation process of molecules, and set the stage, like I said, for MIT, Amyris, and Zymergen to pick that up in 1,000 molecules and build the foundry infrastructure that we have now.
In the last few years, the pace of microbial design, molecule making, and biomanufacturing became furious.
Yes, so by 2019, the goal of making 1,000 molecules with biology was met a full year ahead of schedule.
And the molecule count now, at the end of the project, is well over 1,600, thanks to an integration of automation, machine learning, and other tools for scaling up to this kind of productivity. The gallery of unique micro-made molecules has just been getting richer, and this success has led to a very DARPA thing, to ask yet more of the researchers and collaborators working on the project.
At that point in 2019, we were able to pivot and focus on specific molecules that could be developed further and tested for new DoD operational capabilities. So this meant that now we're looking to produce molecules for DoD test and evaluation partners at scale and based on what kind of testing they needed to do. So think of going from milligram to kilogram amounts of a molecule or material, depending on the application requirements. And as a result of this, the program objectives in this last phase have pivoted away from broad demonstrations of capability, and they've focused more on delivery of DoD relevant molecules in sufficient quantity for testing at DoD facilities.
Participating in this expanded end phase of the project have been performers at Amyris, Zymergen, and MIT Broad, with collaborators in Navy, Army, and Air Force service laboratories. They have been taking the molecule making and materials producing technologies, which performers delivered during the ATCG and 1,000 molecules phases of the project, for test drives. We are talking here of stuff like tough materials for aircraft parts and higher performing liquid fuels. Here's some audio evidence that this isn't just talk. That intense white noise is coming from a test burn of an exceptionally energetic biosynthetic jet fuel. It was manufactured in fermentation tanks by yeast genetically engineered by researchers at Amyris. The test burn of the fuel, which normally would have been made with petroleum feedstock, not yeast cells, took place in China Lake, California, at the Weapons Division of the Naval Air Warfare Center, known as NOCWD.
So we are currently actively working with our partners at NOCWD and AFRL to advance the testing of some of the molecules that we delivered towards the end of the program.
Sunil Chandra is head of research and development at Amyris and oversaw the work that led to this new fuel.
The bottom line is that NOCWD and AFRL see a lot of potential with some of these molecules that were previously not available in meaningful quantities via any other source. And we have been able to deliver larger and larger volumes of these molecules for additional testing in advanced applications. So for example, the NOCWD team created a new fuel that they call Biorenewable 1 or BR1. Which has, I think, close to 19% higher volumetric energy density than conventional jet fuel, in addition to other improved parameters. These improvements will result in an increase in the operating range and fuel efficiency for our aircrafts and ships.
The Living Foundries program just keeps on giving, says Ann Cheever.
From my point of view, this is the most crucial and the most exciting part of the whole program, right? Because we are demonstrating the real potential of engineered biological organisms to produce chemicals, materials, fuels. And I think that it really has had a transformative impact on both DoD needs and the wider bioeconomy. So, it's a powerful technology platform. It can be flexibly programmed. You can scale it. You can adapt to changing environments. Biology can self-repair. So, Living Foundries has been able to address development of the technology and infrastructures to really take advantage of this.
Key to the ongoing success and ability to keep building value on the new, hard-won capabilities Living Foundries has delivered has depended on project collaborators who have been putting their own skin in the game.
So the scale-up work and the partnerships with those DoD teams have been fantastic. They were facilitated by both additional funding from DARPA as well as matching funding from those DoD partners. So we're working with groups from Army, from Navy, and Air Force to perform test and evaluation on molecules. And really the goal here is for DoD test and evaluation partners to develop, test these prototype molecules for DoD applications, and then advance them to a stage that established military technology development pipelines could then transition the technology and field it someday.
That biosynthetic fuel that NOC-WD has tested is one of the more mature examples to have emerged from the Living Foundries program so far. The innovation ecosystem and its payoffs now stand a great chance of expanding and diversifying, both within and outside of DARPA.
Yes, it's been great because in addition to the MIT Broad Foundry and all of their great work, they've published a lot of that work. We were also fortunate to have 2 companies as performers, and so they serve as direct commercial transition partners, and they can incorporate technologies developed under Living Foundries into their own commercial production pipelines. And so we see that as a great sort of win coming out of this program. There have also been some examples of spin-off companies. For example, there was one focusing on biosynthetic pathway design. And we are also proud to incorporate the Embedded Entrepreneur Initiative to help some of our academic performers look at potential commercial applications for the materials that they've developed under Living Foundries.
The Embedded Entrepreneur Initiative, or EEI, is a new DARPA effort. Its mission is to help researchers and innovators with promising but early-stage technology development ideas survive the proverbial valley of death. That's the R&D phase when most startup efforts fail to get to the finish line before managerial, technical, or financial obstacles dry them up and take them out. Meanwhile, also helping the cause is a commitment throughout the Department of Defense to transfer Living Foundry technologies from the lab to operational status. Last year, for one, during the Biotechnology for Materiel and Defense Symposium convened online by the National Defense Industrial Association, Michelle Rozo, then Assistant Director for Biotechnology at the Office of the Undersecretary of Defense for Research and Engineering, said, quote, "The same core competencies that can unlock products and capabilities have the potential to transform military systems and mission spaces."
Yes, there are combined efforts by a wide range of DoD agencies and service branches to develop technology transition pipelines into the DoD and to adopt these emerging biotechnologies like this. So I think, the capabilities developed under Living Foundries and really just biologically based platforms are really going to be able to provide new materials and capabilities for the DoD and the nation, frankly.
For Ann Cheever, Living Foundries, in its completion, is just getting started.
So I think that, post Living Foundries, the breadth and throughput of products will only continue to expand, especially since commercial applications of this technology span across the biotech sector. And for the DoD, this will include continuing that development path. There's going to be new solutions, there's going to be new enhancements to military needs, as well as capabilities.
And that sounds like a textbook example of a DARPA win. Thanks, listeners, for sharing this time with me. I hope you join us again for the next Voices from DARPA. Thanks also to Tom Shortridge for his partnership in producing this program. For more information about the programs that DARPA's Biological Technologies Office now is running, as well as about all of DARPA's other projects, visit darpa.mil. And for links that enable you to download this podcast, go to the Voices from DARPA page on DARPA's website.